Hi, I'm Jennifer Gottwald, a licensing director at WARF.
And I'm IP Manager Victoria Sutton. For the 2020 WARF Innovation Awards, we're pleased to nominate Drs. David Plant and Rita Leash for their work to better understand and quantify sleep.
In collaboration with the company partner, the test they are developing would be the first of its kind. It could not only help to identify and treat sleep disorders, it could also be used for occupational safety and to promote healthy lifestyles.
Congratulations, David and Reed. We're excited to see you take this technology to the next level.
Hi, I'm David Plant. I'm an assistant professor in the Department of Psychiatry, and I'm the medical director at Wisconsin Sleep. Sleep is a really important aspect of all of our lives. We spend about a third of our lives sleeping, and it plays a key role in our overall health. One of the challenges in studying sleep is the ability to measure it. And usually we have to rely on very complicated and expensive laboratory tests that use brainwave activity to measure whether or not someone is asleep. So what this technology fundamentally does is it tries to use an easily accessible sample, saliva, to estimate the amount of sleep people are getting at night and also how sleepy they are during the day, and to do so using changes in genetic expression.
We start with saliva and we extract DNA to monitor an epigenetic modification that is found on the DNA. This epigenetic modification is variable, and it's related to the amount of sleep that a person needs. We have a real opportunity here to take an accessible tissue and very quickly determine the level of sleep that an individual needs or the level of sleep that that individual has been getting. And we can relate this to the health outcomes of that individual.
So we're very excited about further advancing this technology because it has really immense implications for a lot of different industries. So being able to identify how much sleep someone is getting or how much sleep they need has a lot of very practical implications, both for healthcare, because sleep is such an important part of our overall health, and also occupational settings, where knowing how much sleep someone is getting before they operate a motor vehicle or an airplane, for example, is really key to keeping everyone safe. Really, my hope for this technology is that it will be a game changer in the practice of sleep medicine. I'm a sleep medicine physician, and I really think that it's important for us to try to advance our field to care for what is really an unmet need in our overall health and healthcare. So I think having this technology that would be widely available and easy to use, it really could make caring for patients, both in the sleep clinic and then in other settings, much better.
Hi, I'm Leah Hammond, an Intellectual Property Manager at WARF.
And I'm Emily Bauer. For this year's WARF Innovation Awards, we're excited to nominate a cross-disciplinary team working to bridge synthetic biology and electronic communication.
Professors Megan McLean and Bhuvna Krishnaswamy are developing next-generation biosensors with applications from healthcare diagnostics to environmental testing to food safety.
Megan and Bhuvna, we are thrilled to recognize your team's ingenuity, and we look forward to your next steps.
My name is Megan McLean, and I'm an assistant professor in the Biomedical Engineering Department. So there's been tremendous progress in the past decades in engineering sensors through synthetic biology and bioengineering to sense a variety of compounds that would be of interest in health in terms of sensing molecules that signal disease or in terms of sensing what's happening in the soil for better agricultural applications. But it's very difficult because cells themselves are not great processors to take that power and hook it to sort of things that would be traditionally useful. Yeah, so at the highest level, the goal of this technology is to allow biological sensing networks to communicate with electronic networks so that each can do what they're best at. So in biology, we have very good sensors for sensing molecules and other compounds of interest, and then we're very good at engineering electrical networks to perform computation and processing. So the idea is to allow them to talk to each other and each to do what they're best at. So on my end, my lab specializes in optogenetics, which we use to control the activity of these biosensors. And so we've engineered a number of optogenetic tools that allow us to control these sensors with light. So optogenetics takes advantage of naturally occurring light-sensitive proteins and then engineers those proteins so that in response to light, they set off reactions or processes inside of a cell.
So my contribution is in leveraging the simplified bioelectronic network that we've developed and address a variety of these communication problems, how to read from these sensors and build a network of sensors to collect most amount of data, most reliable data from such a network. We envision this in few years down the line to be incorporated in a wearable system that someone can wear an electronic, a bioelectronic sensor where the sensor is on our body and we turn on it, the sensor with the light and detect some activity on our skin or on our body in real time. So that's where we see this going in the future.
Hi, I'm Janine Bermania, the Senior Director of IP Management and Licensing at WARF.
And I'm Stephanie Whitehorse, Director of Intellectual Property for the Physical Sciences at WARF. We are pleased to nominate Professor Kerry Forrest and his team for this year's WARF Innovation Awards.
Their system for generating fusion neutrons could make a significant impact in the production of medical imaging isotopes. It could also play a role in solving the critical challenge of nuclear waste remediation.
Congratulations, Professor Forrest. We are excited to partner with you and your team on this cutting-edge fusion technology.
I'm Kerry Forrest. I'm a professor in the physics department. So in recent years, it's become clear that neutrons are extremely valuable both for industrial purposes and academic purposes. And making neutrons is hard. Normally we would use nuclear power plants and reactors to produce neutrons, but it would be great if we had smaller, more compact sources that could be moved around and were easier and cheaper to build. So we've invented a technique for making neutrons with small, compact devices using an old idea, which is a magnetic mirror plasma trap to confine the fuel for nuclear fusion that makes neutrons. Yeah, so neutrons are useful for many things. Currently in Wisconsin, neutrons are being used to make medical isotopes. There's an isotope of molybdenum 99, which is used in medical imaging. Neutrons are also could be used to burn up nuclear waste from fission reactors. So fission reactors produce waste. If we can make neutrons that have the right energy and enough of them, we could irradiate the waste that comes from nuclear reactors and incinerate it, make it go away so that it can be buried safely without a long-lived effects. We also hope to be able to use the same technology to actually build a nuclear fusion power reactor to make power. We've done the numerical modeling and theoretical modeling to show that it would work on paper. And currently we're building a prototype of the reactor that will be used to demonstrate that the technology actually works.
Hi, I'm Beth Werner, Director of Life Science Intellectual Property at WARF.
And I'm Andy Datina, Director of Licensing. We're pleased to recognize the cancer immunotherapy work of Drs. Jenny Gumpers and Donna Bayou for this year's WARF Innovation Awards.
By combining two kinds of cells found in the body, Jenny and Donna's work could open up a potentially transformative new approach to immune oncology. one that is faster and more specific than current methods.
Congratulations to your great team, and we're very excited to follow the progress in the days to come.
Hi, my name is Jenny Gumpertz. I'm a professor in the Department of Medical Microbiology and Immunology at UW-Madison. We all know that cancer is a tremendous problem in our society, and in recent years, there's been a lot of excitement about using the immune system to fight cancer. So the current ways that we do that have some problems associated with them, and those have to do with not always activating immune cells specifically enough, or in some cases, it's possible to generate immune cells that we actually grow in the lab and genetically modify and put back into patients to fight their cancers. And that's really very exciting, but the problem is that it takes a long time and a lot of cells, and cancer patients just don't have a lot of time. Our invention involves taking a population of cells that we all have and preparing them ahead of time in the lab. Then we can conjugate those cells. We can join them to another kind of cell that's very specific and specifically activates T cells to kill tumors. These conjugates, these pairs of cells, can be prepared ahead of time and stored so that when patients come in ready for immunotherapy, they can be given these cells without delay. So our current stage is that we're testing our human cells in special animal models where we can create a human cancer in the mice and test our human cells in those models. The next stage will be to move on to doing pilot clinical trials, and that will likely take a few years.
During my career, I worked in the fields of immunology and cell biology, and my research focused mainly on basic research problems. I'm really excited that this research has such a high potential to be translated into a clinical trial in the near future.
Hi, I'm Andy Deteen, a Director of Licensing at Wharf.
And I'm IP Manager Victoria Sutton. For the 2020 Wharf Innovation Awards, we're excited to nominate Dr. Randolph Ashton and his team for their work developing improved stem cell-derived models of human neural tissue.
This technology has tremendous potential. It provides a critical step towards bioengineering reproducible spinal cord and brain organoids with applications such as disease modeling, drug development, and regenerative medicine.
Congratulations to Randy and his team. We're pleased to recognize your research in this vital area and look forward to your next steps.
So my name is Randolph Ashton. I am an associate professor of biomedical engineering, and I work over at the Wisconsin Institute for Discovery. I'm also the associate director for the UW-Madison Stem Cell Regenerative Medicine Center. For about a decade now, scientists have been developing methods to culture human stem cells outside the body, and they culture them as a spheroidal aggregate. And when you do that, the cells spontaneously start to differentiate and undergo processes that mimic early human development. And so they can start to create structures that look like what we see in developing human brain, lung, and gut tissues, for example. And so our technology basically allows a very simple method to control this development, particularly for generating models of the brain and spinal cord. And so very early in human development, you form a tube-like structure, which we call the neural tube. And our technology allows us to essentially biomanufacture these neural tube-like structures at will. This will allow us to create models of the human brain and spinal cord that are anatomically similar to what we see in the developing human embryo, and really will allow us to push the technology of creating these types of brain and spinal cord models in a dish to the next level so that they really closely model what normally forms in the human embryo. And that allows us to be much more precise in the therapies we can develop for the diseases that we can now model with these tissues.
We would hope that this technology gets used broadly. Sometimes techniques like this are thought of as being niche because they're so hard to access, but they have such great utility for generating novel therapies that we think the easier this is to use, the more potential we have to, say, affect human health outcomes in a positive manner.
Hi, I'm Brian Freshour, IT Associate at Wharf.
And I'm Licensing Associate Michael Carey. For this year's Wharf Innovation Awards, we're thrilled to nominate a team from the Department of Civil and Environmental Engineering.
Inspired by climate change, Professor Bu Wang and Raghavendra Ragapani have developed a process that utilizes industrial waste such as coal ash for carbon capture and sequestration. Their process is greener with lower energy costs than other methods.
Congratulations to your team. We're excited to partner with you as you work to address one of the greatest energy challenges of our time.
Hi, I'm Bu Wang. I'm an assistant professor in the Department of Civil and Environmental Engineering. The problem we try to solve is how to make carbon capture commercially viable. So carbon capture is this process to remove carbon dioxide from air or from an emission stream, such as flue gas from power plants. So it's an important part of the solution to the climate change issue caused by carbon emission. So particularly, if we can remove carbon dioxide from air, we can essentially turn the clock back, so to speak. However, the problem with the current carbon capture technology on the market is they use a lot of energy and therefore are very expensive to operate. So this technology is to capture carbon dioxide from air or from a concentrated emission stream at minimal energy cost.
The technology we are talking about is a multi-step process that involves mineral carbonation step followed by carbon dioxide capture step. In the mineral carbonation step, we actually generate alkaline solution that can scrub carbon dioxide from air or from a point of source from the industry without the need for any pressurization, and hence has very low energy intensity. So the technology what we are proposing uses an aqueous solution that is fully recyclable, and it does not need any heat energy input, or it does not need any pressurization required. Hence the energy costs are low. The impact is mainly in addressing the climate change challenges, and given the significant amounts of alkaline industrial waste that are generated, we believe the technology can be useful for many industries like power plants, steel plants, even using recycled concrete.
So the next step for us is to build a benchtop scale demonstration unit. This will allow us to demonstrate technology, but also optimize our operating parameters. After that, we plan to conduct some preliminary market discovery and research, really figure out what our customers are going to be and our business model.